Okay. No, so this is the lip, this is the lip, this is the trichrome staining. Trichrome, it's a kind of differentiating staining, differentiating dye. That means the connective tissue fibers are well presented, and nuclei are darkly stained. Connective tissue, I mean the collagen fibers, usually bluish, appear in a bluish color, and the muscle tissue is red. And the epithelial tissues are also more or less reddish, appear in a reddish color. So when we look at this section, this is eventually a just a sagittal section of the lip. We can distinguish three different surfaces. So so this is that surface. This is the mucosal part. Sorry, this is the cutaneous part. It's really easy to recognize because we can see these circular or conical shaped structures. They are the hair follicles. So this is the cutaneous part. Okay. Just a second. And then the other one, the upper side, that is the transitional zone. Also called transitional part. The transitional zone represents just slightly keratinized epithelium, but we are gonna zoom it later. So this is the transitional zone. It's a well vascularized area. So this area, this is it. So eventually the real lip is the transitional zone. So it's well vascularized, and there is a much more thinner keratinized stratified squamous epithelium. And the other one, the irregular shaped posteriorly, that is the mucosal. This is the mucosal, just facing with the teeth, the lower teeth and upper teeth. It depends on the lip. So this is that's the one is the mucosal part. Okay, so cutaneous part or cutaneous surface, transitional part, and mucosal surface or mucosal part are presented. So on the mucosal surface, we can find stratified non-keratinized squamous epithelium. The others, because of a kind of skin covering, we have only stratified squamous keratinized epithelium. And in the core, so eventually this area, here we have the orbicularis oris muscle. You know, it's a mimetic muscle, and it's built up by striated muscle fibers. So that side, it's a cutaneous part having hair follicles. The upper part, just a thinner, well-vascularized area, but there, the mucosal part, it's stratified non-keratinized squamous epithelium with some glands here, but we are talk about later. And at the core, we have a muscle. This is the fibers. Here we can find the fibers of the orbicularis oris muscle. Okay. Yeah. Now let's zoom it. So first the cutaneous part. So these circular structures, these are the hair follicles, and associating to the hair follicles, we have glandular tissue there. This, everything. What kind of glands are these?
Sebaceous glands.
sebaceous glands, so holocrine type of glands. You know, they are associating to the hair follicles, so showing holocrine secretion. So just looking at this is a nice area. So just looking at the epithelium, beautifully seen. Here we have the stratum basale, stratum spinosum, and here we have some granules. So this is the stratum granulosum. You know, as a thin skin, we cannot identify the stratum lucidum, and then there's the stratum corneum. So the typical layers of the stratified keratinized epithelium are presented. So this is the mucosal part. And you remember I said the trichrome stains the connective tissue fibers, and usually they appear in bluish color. So these fine structures, these are fine reticular fibers and collagen fibers, but the coarse collagen bundles are a little bit deeper. So here we have, so these purple, these purple and bluish structures are typically collagen fibers, and the nuclei appear in red color. So these are the fibrocytes mainly. Red blood cells, the red blood cells appear in orange color. So this is a small vessel, and here we have the red blood cells inside. Okay, now that was the cutaneous part. Definitely, just looking at this area, here we have the connective tissue, mainly adipocytes. So here we have, you know, it's a typical, this is a nice one. So here we have the places for the adipocytes, but it's after the routine histotechnique, the fatty components are dissolved. Therefore, we can't see the fat, the exact fat, which we can just see the exact place for the fat, so the place for the adipocytes.
What was the blue stained thing?
The blue, connective tissue, collagen fibers. These blue and purple are collagen fibers.
Okay.
Okay, and then just moving to the core. So this is it. Here we have in the middle. This is typical cross section of the connective of the striated muscle. So here we have the big muscle fibers surrounding a thin reticular fiber, and the nuclei, and that's better. The nuclei. So here we have the nuclei of the cells. They are multinucleated and are located underneath the cell membrane. And that was a really good. So this is a vessel. This is a vessel. So here we have the endothelial cells. There's the adventitia surrounding it, endothelial cells, and there is the preservated plasma. And here we have red blood cells and white blood cells.
Dr. Katz?
Yes?
Can I ask you, before you showed some nuclei around the striated muscle, which nuclei are they?
There?
Yes.
The nuclei of the striated muscle fibers.
Okay, thank you.
You know, they are located underneath the cell membrane.
Okay.
And they are multinucleated, so more than one.
Okay, thank you.
You're welcome. Okay. Then, so second part. Just looking at this, it's a thinner epithelial covering we have, slightly keratinized. So at the top we can find some dead layers. So look at that, but it's much more thinner than the normal corneal layer. We have a really prominent stratum spinosum layer, but at the bottom we can find the stratum basale cells, and then the stratum spinosum here. So it's stratum spinosum, and you know, the lower part, the dermal portion called dermal layer eventually underneath the epithelium represents papillae, and these papillae just invaginate into the upper layer. So just looking at this and that, so these kind of circular structures with vessels, they are because of the section, the wavy line section of the dermal papillae. So they are the invaginated parts of the lower portion, the lower layers. Okay, so it's these kind of weird things. It's because of the section they relate to the connective tissue underneath. So stratum spinosum again. Here we have some granules here. So we have a really thin layer of stratum granulosum and no lucidum, and there is a really thin corneum. But don't forget, eventually this area is just for the transitional site between the real cutaneous layer. So here we have, and at the top we can see the detached keratin layers, and it's also a kind of thicker area. So here we have normal or more or less normal thickness of keratin layer, but then just going toward the posterior side, it's getting thinner and more thinner, and eventually here, so we can see again, we can see now here we can see the exact corneal covering. We have mainly spinosum and granulosum. So this is the real transition between the stratified non-keratinized and keratinized layer. Okay, let's see the mucosal part here. So it's a typical stratified non-keratinized squamous epithelium, and it's 3 layers here. So here we have the stratum basale, there beautifully seen. Over it we have the stratum spinosum, and the flattened compressed cells form the stratum planocellulare. This is the planocellulare. So this is a typical stratified non-keratinized squamous epithelial covering. Then we also have dermal papillae here, so there underneath the layer, but no hair follicles. Just looking at this area, we cannot find the typical elongated conical shaped hair follicles. What we have, these glandular portions, mainly mucus acini are presented, so called labial glands. So labial glands, these are the labial glands, mainly mucus glands, but among the mucus glands, we can find some serous ones. Okay. So that's better because here we have mucus glands as well. So look at that.
Are those intercalated ducts next to them?
Wait. So here we have the mucus. The nuclei are compressed and flattened to the bottom. We can see the lumen of it. And this is a serous. This is a serous. There is another one, but it's a little bit better. And look at that. It's really nice. So here we have the acinus, the serous acinus, and you see the nuclei of the cells and the exact lining cells become short cuboidal. So here we can see the beginning, the exact beginning of the intercalated duct. It just leads from the serous acinus. So it's really nice aspect. And the majority of the ducts, I think it's also it's an intercalated. There is an intercalated one, so it's short cuboidal. There's the preservated juice inside. This is a striated duct, but this is much more thicker. So we can say it's interlobular. But here we have nice genodidamylone. So here we have there and there. And there is a kind of a longitudinal section of a small capillary with endothelial cells and red blood cells. So I think it's a kind of easy slide. Just the surfaces are important, and at the core we have the muscle, the orbicularis oris muscle. So cutaneous part, transitional part, and mucosal part. It's a trichrome stained section, therefore the connective tissue elements, the fibers appear in a bluish or purple color. The cutaneous part represents hair follicles covered by stratified keratinized epithelium. The transitional zone is a much more thinner area covered by
Hello?
Yeah, we can hear you.
Yeah, we lost him.
I can't hear. He left, right?
Hello, hello. I don't know.
I think
No, we lost him.
Yeah.
Yeah.
Maybe he'll make a new meeting because he's not the host anymore, so he left.
Oh.
So, what's up, guys?
How was the test for you guys? It was good.
Chill.
It was actually really good, yeah.
Yeah.
Except question number 5. I don't know if you guys know the answer.
I didn't know the gag
Which
question.
one?
Me neither.
Yeah, the gag stuff.
Yeah, the gag was.
I just guessed hyaluronic acid because I saw it like
Yeah.
everywhere.
Yeah, no, because like, all
Should have
the
gone for it.
all the other answers were with phosphate or something. So like this was like the only unique answer. So I also the rest of the answers were like phosphate, something phosphate, something phosphate.
Androgen phosphate?
Yeah,
Nah, I choose this one and it was wrong, so.
yeah, but like no, like I put the acid one.
Oh, hyaluronic acid?
Yeah, I don't know. I saw everything phosphate, phosphate, and one answer was like with acid. So I thought like.
I'm gonna email Dr. Katz and ask him what we should do. I think maybe we should all leave the meeting and then he'll
Yeah.
organize a new one. Yeah, that's
Yeah, maybe he's still talking.
I think I'll just leave and everyone should leave and then he'll just organize another meeting.
Maybe
Okay.
he's coming back though. Maybe he's trying to come back.
I don't know if he could join.
Yeah, you need to be the
Join.
host.
We just lost the connection.
Whoever Omar is should like close the meeting.
I will be the host. Okay, I'll invite him. Who can I?
No, but you can't.
If you can email him, I will tell.
Or shall I just wait?
Okay,
for
I
the
just emailed him. Okay, great.
Okay, thank you.
Yeah, don't leave yet, because if he comes back,
Yeah, he probably just lost his
Don't
connection.
worry, I'm not going anywhere.
okay. I just hope he knows that he froze, because maybe he doesn't even realize it.
Oh, he's
my-
the host now.
I hope not.
He's
He's
back.
back.
Here it is back.
Yeah, hello. Something happened. The Zoom just freezed.
Yeah, we lost you for a moment.
So it was interesting. No. Just a second. I open again the case viewer. Okay. So let's see our next slide. The tongue. The tongue. So first, related to this topic. So you see, here we have the okay. So here we have the tongue. The surface of the tongue eventually lined by stratified non-keratinized squamous epithelium. It's not unusual because you mentioned it many times. So in the oral cavity, in the part of the esophagus and upper part of the pharynx, lined by stratified non-keratinized epithelium. In the tongue, eventually, we can find numerous striated muscle fibers. This is a typical specific one called visceral striated muscle fibers, so visceral striated, because they are under voluntary control, but they are not connecting bones to each other. So eventually we have muscle fibers and glands related to the surface and related to the deep areas as well. So onto the surface of the tongue, we can find mucosal projections called papillae. Papillae. We have 4 types of papillae. The most common one is the filiform. So the filiform papillae, they are conical shaped structures representing highly keratinized. So it's really weird because we said the tongue is all in generally non-keratinized epithelium, but the filiform papillae represents at the top of them highly keratinized epithelium. Therefore, eventually this epithelium determines their function. They have only mechanical role, just increase the friction between the tongue and the food bolus. So this is the main function of the filiform papillae. They don't represent taste buds, so no taste sensation onto the surface of the filiform papillae, just they have mechanical role, increasing the surface and increasing the friction between the tongue and the food bolus. Okay, so these are the filiform papillae. And then the next type is the fungiform papillae. They are typical mushroom-shaped papillae, mushroom-shaped mucosal projections. At the top, we have stratified non-keratinized epithelium, and some taste buds at the top of the fungiform papillae are also presented, but eventually the fungiform papillae are really typical and well developed in the dogs, so not in the humans. So we have just a few fungiform papillae, and just a few taste buds are presented at the top side of the fungiform papillae. Now let's see our first slide, which is the 34. Yeah, this is it. So this is a cross section of the tongue, and at the top of the surface we can find these mucosal projections. Inside we have muscles. So look at that. You remember when we studied the tongue, we mentioned longitudinal muscles. We have superior and inferior longitudinal muscles. Superior muscles are there, inferior here. And among them, we can find the transverse and vertical muscles. So these are the transverse and vertical muscles in this area. So superior longitudinal right underneath the epithelium, inferior longitudinal here, and among them we can find the the transverse and vertical muscles. So let's see the structures. I think it's more prominent here. This papilla. This is a typical mushroom-shaped papilla. This is a fungiform papilla. The connective tissue eventually invaginates into the projection into the papilla, and onto the top of the surface we can find stratified non-keratinized squamous epithelium. Unfortunately, we can't see taste buds, but there is another one. Yeah, it's also a mushroom shaped. So here we had, but it's a little bit elongated as well. So here at the top side, there is a taste bud. This is a taste bud. So this is a fungiform papilla, but this is a typical shaped one. The others, so these papillae, so these all are eventually filiform papillae, and looking at the top side, here we have stratified keratinized epithelium, just covering the filiform papillae, just increasing the friction between the tongue and the food bolus during the swallowing. Okay, and then underneath them, I just want to show the typical cross section of the striated visceral muscles. So here we are lucky because here we have longitudinal and cross section as well. So beautifully seen on the cross section. The nuclei are underneath the cell membrane. They are multinucleated, but if we look at the longitudinal section, we can see beautifully the striation. And among the muscle fibers, definitely we have connective tissue fibers, collagen fibers onto their surface. Numerous fibrocytes are there, vessels are there, so everything related to the connective tissue. Okay, so two papillae, filiform, fungiform papillae, only the fungiform from these two have taste buds, represents taste buds for the taste sensation. Now let's move on. Our next type of papilla is the foliate papilla. The foliate papilla eventually a little bit bigger than the elongated filiform papillae. At the top we can find stratified non-keratinized epithelium, but at the lateral side we can find these circular cabbage-like structures. They are the real nice appearance of they are the taste buds in a different location than in the fungiform papillae. So in the foliate we can find the taste buds at the lateral sides, and between two foliate papillae we can find deep clefts, eventually also lined by stratified non-keratinized squamous epithelium. And looking at the foliate papilla, we can find this kind of W-shaped invaginated connective tissue. So this is really typical for the foliate papilla. Or there is another aspect. If we just check the surface, we can say it's M, but here we can see the exact W from the connective tissue side. So I think it's beautifully seen. Okay, so this is the foliate papilla. So for the foliate papilla, we have the number 50. So here we have, this is from the monkey, this is from the human. They're not so nice. Okay, let's see the monkey first. Oh, that's much more better. So this is a foliate papilla. This is a foliate papilla. Here we have this W-shaped connective tissue from the dermal area, and these small circular structures, these are the taste buds. Oh, this is a beautiful. So look at that. Look at that. This is a circular structure. Here we have the entrance of the taste bud called pore. This is the taste pore, and the main cells inside, the big nuclei are related to the neuroepithelial cells. So they are the exact sensory cells. They eventually sense the components of the saliva, because the saliva dissolves the taste molecules, and eventually the saliva just wash these kind of dissolved molecules into the pore in the taste bud. So the big nuclei are belonging to the sensory cells. They are modified neuroepithelial cells. The slender nuclei, the flattened ones, are belonging to the supporting cells, just among the sensory cells. And at the bottom of the taste buds, we can find basal cells. They are kind of stem cells. Just they differentiate into supporting, either supporting or sensory cells, so they are stem cells. So in the taste buds, we can find 3 types of cells. We have the sensory cells with the big round shaped nuclei, and the elongated nuclei belonging to the supporting cells. And the basal cells are stem cells, just differentiating into either supporting or neuroepithelial sensory cells. Okay.
Dr. Katz,
Yes?
how do you tell the difference between the supporting cell and the basal cell?
The nuclei. The basal cells are at the bottom and smaller, and the supporting cells nuclei are elongated and a little bit bigger.
Ah, okay, thank you.
There is it's not really nice, but wait a second. So here we have, this is another surface. These small cells, this nucleus belongs to the basal cell. These elongated ones, part of the supporting cells, but these huge ones are belonging to the sensory cells. So supporting, long, elongated, basal cells, and the nuclei of the sensory cells.
Dr. Katz.
Okay.
Sorry?
Dr. Katz, why are the nuclei of the basal cells, not the basal cells, of the sensory cells different color from the
because it's just for the sensation, just transmitting the impulse. The supporting cells eventually have more active production or more active synthesis inside.
Okay, so it doesn't have to do with something that they contain.
No.
For example,
No,
okay.
no.
Well, the basal cells are more purple because they contain more protein, no?
Yeah. So it's more so it's darkly stained because of the really active cellular functions. Okay, so they were the foliate papillae. Now, so that was the monkey. Now I'm gonna see it again, the human. Where is the where is it? That's the one. No, they are not nice. We can see the taste buds. Okay, so the monkey was perfect for this, for the foliate. Now let's see the last type of the papillae. These are the circumvallate papillae. Eventually in human we have 8 to 12 circumvallate papillae. These are the biggest papillae. They are located just in front of the terminal sulcus. You remember the terminal sulcus? That is a V-shaped structure, and then the tip of the terminal sulcus we can find a developmental remnant of the foramen cecum. You know the foramen cecum, it was just the remnant of a tube. It's a developmental tube, and it eventually just for the descent of the thyroid gland during the development. So at the tip of the V-shaped terminal sulcus, we can find the remnant of the thyroglossal duct called foramen cecum. So in front of them we have huge circular structures called circumvallate papillae. They are embedded into the surface of the tongue. Therefore, we have a huge deep cleft surrounding them, also called a moat-like invagination around the circumvallate papillae, and the lateral sides of the papillae represents the taste buds. Numerous taste buds are there. So, and there is another thing related to the bottom of the deep cleft. We can find some serous glands, call them von Ebner's glands. So von Ebner's glands are related only the circumvallate papillae, and eventually the juice of the von Ebner glands just continuously wash these really deep moat-like invaginations surrounding the papillae. Now let's see this papilla. This is the 49. So these are, so on the small magnification, we can see this and this. These are circumvallate papillae. They surrounded by really deep moat-like invaginations. So, and then we just zoom in. At the top of the circumvallate papillae, we can find the typical stratified, non-keratinized squamous epithelium, and then underneath we have just connective tissue with collagen fibers, fibrocytes. When we look at the lateral side, we can find these eventually disrupted structures. These were previously the taste buds, but this one, it seems intact. So this is a taste bud. This is a taste bud, and here we have the pore, the taste pore, and these cells, bigger cells, the paler ones, are belonging to the supporting cells. A little bit darker cells, part of the belong to the supporting cells, and at the bottom, maybe these are the basal cells. These are stem cells. Okay, stem cells, basal cells, supporting cells, these, and sensory cells in the taste bud. And here we have this huge invagination, and underneath these moat-like clefts, we can find the serous von Ebner's glands. So they are typical serous structures. The serous acini are presented there, so everywhere they are serous acini. So these are the von Ebner's glands. So looking at this aspect, these are the serous ones. Only the serous ones are von Ebner's glands, because I said, you remember, in the tongue also we can find some another usually mixed glands. And just zooming in this area, here we have mucus. They are mucus, or mainly mucus glands. So these are not the von Ebner's glands. The Ebner glands, the von Ebner's glands, are serous glands, and they eventually secrete juice, cleaning the moat-like invaginations surrounding the circumvallate papillae. Okay, now let's move on. Let's see the glands. Yeah.
I just wanted to ask, so the only ones that have taste buds are the fungiform and the circumvallate?
And the foliate. We check the foliate as well.
This last one that we looked at, was that the foliate?
The last one was the circumvallate.
Okay. Okay, thank you.
You're welcome.
Dr. Katz, I have a question.
Yeah.
So the only difference, or is there any difference between the von Ebner's gland and Serous gland, or is it just called von Ebner's gland because it's found in the tongue? So it's just normal Serous gland.
I haven't heard properly. So eventually the von Ebner's glands are related to the circumvallate papillae only.
Right, but they're normal Serous gland. There's nothing special about them.
No. They are just related to the
Tongue.
papillae. I mean the circumvallate.
Thank you.
You're welcome. Okay, let's move on. Submandibular gland. Our next. Yeah. So submandibular gland. The submandibular gland is a mixed gland. It's not unusual for you. It's not unknown for you because we covered the serous and mucous acini related to the submandibular, and it's surrounded by. So the submandibular glands are located in the submandibular trigones. So they are surrounded by moderately dense connective tissues. When we just zoom in, we can find the fibers here. So this is a capsule around. So here with connective tissue fibers, nuclei, everything, and from the connective tissue capsule, we have septa just separating the gland into lobes. So here we have a beautiful septum there. So this is a lobe here. There is another one, and here this. So these are the lobes. But in the lobes, also from the separator connective tissue, we have another fine connective tissue fibers separating lobules. So we have lobes and lobules. And also we can say we have numerous adipocytes in the submandibular gland as well. And in the capsule, in the septa, we can find large blood vessels, and definitely inside the gland, not just blood vessels, we have duct just carrying the saliva toward the carbuncle. Okay, now let's see the glands. I mean the acini. The submandibular gland as a mixed gland represents serous and mucous acini, but the predominated acini are the serous acini. So more serous acini are presented than mucous acini. So looking at this aspect, here we have the mucous acini, these, but the majority of the acini is serous. So the darkly stained are serous acini, and the typical aspect of the serous acini, they are round-shaped. Inside the cytoplasm, we can find really intense staining of the dye, and when we just zoom in properly, we can find these small granulations inside. These are the zymogen granules. That means inactive enzymes, for example, because this is a salivary gland, so the inactive forms of the proteins are covered by these granules. So zymogen granules are related to the serous acini, and mucigen granules are also presented in the mucous acini. So this kind of foamy appearance of the cytoplasm, we can find the mucigen granules, but they are not so prominent than the granules, the zymogen granules in the serous acini. So serous acini, we have zymogen granules. Mucus acini, we have mucigen granules. And this is a typical aspect of the mucus acinus. Nuclei are flattened, pushed to the bottom. Cytoplasm is foamy, and there is a paled appearance. It's a pinkish paled appearance color of the acinar cells. And then also we can mention some additional cells. So look at that. There is another acinus. By the way, here we have a mucus acinar cells, and here we have serous acinar cells, and we can say this is a really small ganozydomylon. This is a serous cap, but this cell is outside from the acinar cells. But close to it. This is a myoepithelial cells. Myoepithelial cells, the modified smooth muscle cell, or just you can say it's just a cell having contractile activity. Eventually, they just help to push and support the wall of the acini. So this is a myoepithelial cells, modified smooth muscle cells. There is a capillary. Here we have a red blood cell. This is an endothelial cell there. But here we have another really nice myoepithelial cells. So here we have the basement membrane, this pink line. This is the basement membrane for the acinar cells, and this is just like attached to the wall. This is a myoepithelial cell. Here we have another ganozydomylon. Okay, this is it. So ganozydomylon means serous cap. This is the other meaning. Okay, there is another ganozydomylon. So I think everybody knows the ganozydomylons. Duct system. Just zoom in, but it's not really nice. This is it. This is the first part of the duct system, the intercalated duct, where the cells, the lining epithelial cells, are short cuboidal cells. So the nuclei are in the center of the cells, and the cytoplasm is really similar to the staining of it is similar to the mucus acinar cells, so it's paler, not darkly stained. I tried to show another one because it's not really nice. So this is it, that's good. There is it. So it's not a proper cross section, it's a tangential section, but I think it's perfect to get the meaning of the intercalated duct. So comparing with the acini, the real structures, the circular structures, the intercalated duct, the cross section of the intercalated ducts are smaller. So I think it's really important. They are smaller than the acini. The cells are in the center of the cells, and the cytoplasm is pinkish. It's a little bit similar to the mucus acini cell. So, but the nuclei, in the mucus acinus, the nuclei are flattened and pushed to the bottom. But here we have prominent round-shaped nuclei in the center of the cells. We can see a small lumen as well. So this is a typical intercalated duct. This is a typical intercalated duct. Okay. There is another one. There is another one here. But these are also belonging to the wall, but you can see the lumen. The next part of the duct system is this. This is a striated duct. This is a striated duct lined by simple columnar epithelial cells. The cellular borders are beautifully seen. So here we have the borders of the cells. The nuclei are well presented at the so round-shaped structures in the center of the cells. And when we just zoom in the bottom of the cells, somewhere we can find some, so these, some striation here. You know, that's the reason why we call them striated ducts. So that the basal portion of these cells, we can find the wavy line basement membrane having pockets. And in these pockets, we have large amount of mitochondria. This mitochondria is just for the really intense transport through the cells. So there is absorption and a secretion, because the final modification of the saliva occurs in the striated duct, so in the duct system eventually. So at the basal part, we have wavy line basement membrane of the cells having pockets, and in these pockets we have mitochondria. And this kind of arrangement of the mitochondria cause the little striation at the basal part of the lining cells. That's the reason why we call it striated duct. And the other type of the ducts, yes?
Could you please point at the striations again in the pockets? I just wasn't able to see it.
We can see, definitely we can see the mitochondria. When we just zoom it, the cells, we have something there, maybe some lines, some fine lines. This is the striation. But we need a really good light microscope to show them. This is eventually a electron microscopical structure. I mean this striation and the mitochondria. So definitely we can definitely see the mitochondria. It's impossible.
Okay, thank you.
You're welcome. Okay, and the next part of the duct system here, when we have stratified lining epithelium, this, so beautifully seen, it's 2-3 layers of nuclei are there. This is an intercalated. This is an interlobular, sorry. Intercalated was the smallest. So interlobular, this is interlobular duct. Interlobular ducts are also presented in a bigger caliber. So there, this is another interlobular. Here we have, when the epithelium is getting higher and higher, and the columnar is the last one, because I mean the columnar line is the last part of the duct system, because you know the lining epithelium eventually gradually increases and raise toward the surface. So the whole duct system started with the intercalated duct. They are lined by short cuboidal epithelium, and then they become a simple columnar epithelium. They become striated ducts, and then the final part, the terminal part of the duct system, lined by stratified, first cuboidal and then columnar epithelium. So this is a typical stratified columnar epithelium lining an interlobular duct. We have one thing to check, because among the acini we can find plasma cells. I want to show a nice one because it's not so nice. These are plasma cells. So this is a duct. This is an intercalated duct, but it seems not simple lining epithelium, but just because of the section. But here we have the serous acini there. But these cells, this is a typical one, and that is also this is plasma cell. This is plasma cell. They secrete immunoglobulins, mainly IgA, related to the salivary glands. So these are plasma cells, terminally differentiated B lymphocytes. So they secrete the immunoglobulins. I'm gonna show another one. These are also. So look at that. This and these are plasma cells. But look at the nucleus. Look at the nucleus. So it's not a homogeneous because of the huge clumps of chromatin inside, forming the cartwheel-like manner. So or also called clock face. So here we have the huge clumps in the nuclei. They are really typical for the plasma cells, and there is a prominent rim of cytoplasm. So when we identify a plasma cells, we should recognize before a cytoplasm, a prominent rim of cytoplasm, which is also more or less darkly stained, and the nucleus represents huge clumps of chromatin inside, closing a clock face, or also called cartwheel like manner.
Dr. Katz, what about the one right next to it? Is
This?
that the one next to that one?
This?
The one to the left. Is that no, to the left of the one you were pointing at?
That?
Yeah, is that also plasma cell?
Also plasma cell. It's also plasma cell. I can't see for this is plasma because it's too the cytoplasm is too small, and I think it's maybe plasma, but it's not in the exact real section. This is also plasma cell. So these and these and these are plasma cells because of the strongly stained cytoplasm and the nucleus. Okay, now for this we have another slide, the Movat stained. When we can see the mucus ossinae in turquoise color. So looking at this area, we can find these turquoise structures. These are greenish, bluish greenish structures. These are the mucus ossinae among the serous ossinae, but we can see the serous ossinae also presented. So they are also present some turquoise granules because of the carbohydrate components of the cells. So the carbohydrate level, it's really typical for the mucus ossinae, while the serous ossinae are more they secrete more protein-containing juice, but beside the proteins, we have some carbohydrates as well. That's the reason why the Movat, this turquoise color, also present in the serous ossinae cells. So this is the Movat staining, and here we have the duct system. So striated ducts are mainly, the intercalated ducts are more difficult to find on the Movat staining.
Dr. Katz?
Yes?
Is the small reddish colors the plasma cells next to the serous ossinae? Or purple color?
These?
Yeah.
No, these are definitely not. It's impossible to be plasma cells because the plasma cells are not secreting carbohydrates. So they are just part of the wall of a serous. This is a serous ossinus, and it belongs to it. The plasma cells, maybe this is a plasma cell. This is a vessel with red blood cells, and this is a plasma cell. It seems it's part of the ossinus, but it's not.
Thank you.
You're welcome. This is a capillary with red blood cells. This is another plasma cell. So here we have the cytoplasm, a prominent cytoplasm. Here we have the nucleus, and there is a little bit dark area inside. So we have the clumps of the chromatin, so it's a plasma cell. But the hematoxin is much more better to show the structures of the, maybe here we have numerous plasma cells. They are all plasma cells. And you see, you can see these clumps of the chromatin inside of the nuclei. So here we have the cytoplasm. There is another rim of cytoplasm. So these are plasma cells secreting immunoglobulins. So to demonstrate the glands, the hematoxin is the most suitable dye, not the MOVAT. But the MOVAT stains the carbohydrate contents in the acini. Now let's see the.
Dr. Katz, why is it useful to have stained carbohydrates?
It's just because of the different components. So we have so for example the PAS staining, and also we have another musicarmine staining. They are just for the carbohydrate contents. It's eventually just a different aspect.
Okay.
Our next gland is the sublingual. That's the 9. Yeah. Here we have. So this is the sublingual gland, but it's a unique slide because it shows a little portion of the submandibular gland as well. So just looking at the whole gland, you see here we have a darkly stained, it also belongs to it. So the darkly stained area. So eventually, here's the border, there. So this is the right side. The right side is the submandibular, and the left side shows the sublingual portion. And the sublingual gland also mixed gland, but they different because the sublingual glands represents mainly mucus predominance. So the mucus acini are the more prominent structures related to the sublingual. So let's zoom the sublingual. So look at that, just looking at this aspect. In the lobules, we can find mainly this paled structure. These are the mucus acini, and among them we have some serous acini as well, but mainly mucus acini are presented. So these are mucus, and these darkly stained here, these are serous acini. So mainly mucus acini. Here we have adipocytes, definitely, but not so much than in the submandibular. And our next gland, the next main salivary gland, the parotid gland, is the because of the biggest gland, represents large amount of adipocytes. So it's really typical for the parotid. So sublingual gland, mixed gland with predominance of the mucus acini. So here we have serous. Some serous acini are among the mucus acini adipocytes. Eventually, the same duct system are presented. Try to find intercalated duct. It's definitely a striated duct. This is a striated duct, but here we have, this is an intercalated.
Dr. Katz, is the striated more eosinophilic?
More eosinophilic, yes. More eosinophilic than the intercalated as well.
Are we supposed to know why?
Sorry?
Should we be able to explain why?
Because they belong to the secretory or the excretory portion. The secretory portion cells are darkly stained, but the excretory portion components, excretory portion of the duct system, eventually they have less cellular activity than the acinar cells.
That makes sense. But the intercalated duct doesn't secrete, does it? I thought it's only the first part of the duct system.
It's just a tube leading from the acinus. It doesn't secrete, but it's really small and really close to the acini. So don't forget. So it's that's the reason why it's a little bit darker sometimes.
Okay.
Okay. So this is an intercalated duct because you know, this is a typical aspect of the mucus. The serous acini are much more bigger. So here the size of the structure is the key to figure out which one is.
Okay, thank you.
You're welcome.
Dr. Katz?
Yes.
Did you say that the excretory portions were more darkly stained, and the secretory more?
Secretory is darkly stained. Secretory.
Okay.
Not the excretory.
All right. Thank you.
Yeah. Okay. Okay. Let's see the. So here we have, but it's stratified. It's two. I think it's 2 layers. I think now here we can see the transition between the striated duct and interlobular duct, because here on that side we can see only 1 layer of cells, but it becomes 2 layers. So here we can see the exact transition between the simple and stratified lining epithelium. Here we have the preservated saliva inside. But it's not really nice. But here we have 1 layer, 1 lining covering. So this is a striated duct. I tried to show a nice one because it was not so nice.
Dr. Katz?
Yes.
So you are saying that the striated duct becomes the interlocular?
Yes, yes, yes. This is the same tube. Intercalated, striated, interlobular. This is the same tube. This is the same tube, just getting bigger and bigger, and the lining epithelium is getting thicker.
So the places where we see intercalated is just at the moment because it didn't get to the point to become the next. Ah, okay, okay, thank you.
So here we have, this is a striated duct, and there here we have a stratified lining epithelium. So this is an interlobular duct. Also common mistake during the oral exams to mix the vessels up with the ducts. It doesn't matter what kind of tissue we are talking about. So you should always check the lining epithelium, because in the vessels we have always squamous epithelium, only one layer of squamous epithelium, only one layer of endothelial cells. So quite obvious to look at this. This is the duct, and here we have this is a vessel. This is a vessel, and here we can see the endothelial cells, but there is a help. The presence of red blood cells and white blood cells is a huge help, but sometimes we can find red blood cells in the duct system. Don't forget, when we make this section first, we just cut a small cube from the specimen. Therefore, the surfaces are bloody. So eventually, it's the presence of red blood cells. It's not a really big help. Look at that. Look at this side. Red blood cells are just over the adipocytes, but it's impossible. The adipocytes doesn't represent red blood cells, so it's an artificial something. But during the preservation, some of the red blood cells cover the surface of the adipocytes, so it's an artifact. Then let's see another. Just looking at this. So here, this is a really huge duct, and here we have these are white blood cells, by the way, white blood cells, and definitely somewhere some red blood cells are here also presented. These are red blood cells, but look at the epithelium. This is a stratified columnar epithelium, so it's part of the duct system. This is a really huge excretory duct. But looking at this, here we have some red blood cells, but I try to, here we have an endothelial cells in the exact section. So this is a vessel. This is a vessel. Okay, so always check the lining epithelium. It's quite obvious. Here we have stratified columnar epithelium. This is a duct. Okay. Now our last slide represents the parotid. The parotid gland. This is it. Parotid gland is completely serous. So when we're talking about the salivary glands, we have three glands, three bad glands, obviously. So the submandibular, sublingual, and parotid glands. Sublingual, submandibular glands are mixed glands, while the parotid is completely serous. They are surrounded by capsule, dense irregular connective tissue capsule, and from the capsule we have connective tissue septa dividing the glands into lobes, and inside the lobes we can find small units called lobules. And we have the acini. In the mixed glands we have Januzzi-Damilones as well. And all in 3 glands, the parotid, submandibular, sublingual glands, we can find the different parts of the duct system. So we can find the intercalated, striated, and interlobular ducts, but these are belonging to only one tube system, only one structure, just lining, just draining the saliva toward the surface or a lumen. So this is the parotid gland, and there is a unique specimen because in the section we can find a lymph node as well. So this darkly stained area shows the lymph node as well, but you don't need to know the lymph node, we haven't covered yet. So let's focus on the glandular tissue. I said it's completely serous. So look at that aspect. There is another fact related to the parotid gland. Numerous adipocytes are present. It's so more numerous than in the submandibular gland. So these white areas are related to the adipocytes. So here we have serous acini, everywhere serous acini. We have some lightly stained structures, but this is a striated duct. This is another striated, but the smaller ones are the intercalated, part of intercalated ducts. Okay, so they are not mucus acini. This is, by the way, this is the longitudinal section of an intercalated duct. This is a striated duct, simple columnar epithelium, and also beautifully seen the serous acini, the zymogen granules, so these round-shaped granulations inside, so the inactive form of enzymes, for example. But also related to the ducts, we can find myoepithelial cells. So myoepithelial cells are not presented only around the acini, because we can find them around the ducts as well. So this is a myoepithelial cell, and there is another one just tensing, pushing the wall of the acinus there, and then it also helps to push the saliva out from the striated duct.
Dr. Katz,
yes?
A few seconds ago you showed us an intercalated duct. You said it was a longitudinal section of it.
Here.
How did you look at it and know straight away that it was an intercalated duct?
The color, the staining, it's more eosinophilic, you see? So they are darkly stained structures. These are the acini.
Yes, but how did you know it wasn't a striated duct, part of it at least?
Because of the size. It's too small for it. It's too small. This is the wall. We can see the lumen, but this is the striate. This is the intercalated, because it's too small. It's a much more. It would be bigger, like this.
I see. It's just part of it, though. So, okay, I understand. Thank you.
I try to show another one. The parotid is really good for the intercalated and the submandibular, while the sublingual is really difficult to find them. Because sublingual, I mean the section of it, it's not so nice. I mean, it's something, there is a shrinken components. So look at this. This is an intercalated. This is an intercalated duct. Here we have the lumen, here we have the nuclei, and at the center of the cells, paled or lightly stained cytoplasm. So it's a typical intercalated.
Okay, thank you.
You're welcome. Avita, look at that. This is a longitudinal, a wall, just the wall of the striated duct. So it's much more bigger. You see, here we can see the columnar appearance of the cells.
Right.
So this is just a tangential section, just shows the wall of the striated duct.
Okay.
Okay. And it's not so nice. This is the very beginning of the striated duct. Okay. Definitely here we can see a really big interlobular duct. It's lined by stratified epithelium, columnar or cuboidal epithelium. So this is a huge duct. And look at nice. Look at that. Numerous red blood cells are inside, and this is plasma. This is a plasma. So the preservated plasma. So, but this is a duct. It's not a vessel. It's just because of when the pathologist cut the specimen, making small cubes and put it into the formaldehyde. So that's the reason why we can find some blood and red blood cells more or less everywhere onto the surface and in the lumens. Okay. So always check the lining epithelium. Okay, this is a really nice, simple columnar lining epithelium. This is a striated duct. This is a striated duct, and maybe if I can raise the magnification, there are some really delicate lines at the bottom of the lining epithelia cells. So this is eventually the basal striation, but it's rarely seen. Eventually, we need a really good section of the cell and really good microscope to show it. Maybe this, look at that. There. So these really delicate lines are the basal striation. Okay, so looking at the parotid, we should mention first it's a completely serous gland that having serous acini only, large amount of adipocytes, and related to the duct system, we can mention the intercalated, striated, and interlobular ducts. And definitely, I haven't mentioned the plasma cells, but we have plasma cells here as well among the acini and close to the, this is it, close to the cells, close to the ducts here. This is a plasma cell. Okay, so there is a more or less darkly stained cytoplasm. There is and the cytoplasm is well presented. And look at that, at the periphery of the nuclei, we can see the clumps of the chromatin. This is a typical plasma cell. So in the salivary glands, we can find numerous plasma cells, okay, secreting immunoglobulins. Okay, so I think